Hi everybody. For those of you that don't know me, my name is Louise Lavictoire and I'm the Interim Head of Science at the FBA. Welcome to the latest instalment of the FBA seminar series. We're exploring the careers to date of the FBA Fellows and telling you a bit about the key projects that the FBA is involved with. So I hope you've enjoyed the seminar series so far. If you've missed any of them, you can still catch up with them on the FBA website. I'll put a link in the chat box for you now. So just to let you know that these presentations are all brought to you free of charge. So if you're not an FBA member and you'd like to support us, you can do that under the Join Us tab on the FBA website. So that's fba.org.uk. So without further ado, I'd like to welcome Dr. Steve Brooks with us today. So Steve started working at the Natural History Museum in London in 1979, where he curated and researched freshwater insects and lacewings. He later focused his research on the use of chironomid midges as quantitative indicators of environmental change. He's author of over 250 research publications and has written several books on dragonflies. He's a member of the Conservation Committee of the British Dragonfly Society and is chair of the Riverfly Partnership, which is hosted at the FBA. He's a scientific associate at the Natural History Museum following his retirement in 2017, and of course is one of our FBA fellows as of 2021. So I'd like to invite you to take it away, Steve. If anybody has any questions during Steve's talk, put them in the chat and we will ask questions at the end.
Okay, well, thanks, Louise, for that kind introduction. Let's just get this talk up. Hopefully everybody can see that. Start it. Okay, so as Louise said, I'll follow the tradition of talking about basically what my career was about and how I got there and perhaps where I want to go now. I've retired from the museum. So as some of you may know, I've spent decades really working on chironomid midges, and I've come across a lot of people not really knowing what midges are, and even people who do know what midges are really treat midges with disdain. So what is wrong with midges? Well, most people, when they ask me what I used to work on and I say midges, they say, oh, good. Have you been working on wiping them out from Scotland, where they spoil our holidays because they bite us? Well, of course, as all members and fellows of the FBA know, chironomid midges don't bite. But nevertheless, even fellows of the FBA are pretty unimpressed by midges. They say they're uncharismatic, they're hard to identify, they're boring. What use are they? Well, some people use them as food for their tropical fish, or dragonflies like eating them. But actually, I hope by the end of this talk, I'll have persuaded you there's a little bit more to midges, and maybe persuade some of you to start a career in midges, or at least end your career looking at some midges. So where did it all start? Where did my interest in freshwater ecology and so on start? Well, I've got this photo here of me in Devon in 1959, when I must have been about 4 years old. And you can see I'm looking under a stone in a stream. My dad was really interested in freshwater. He studied zoology at Bangor University. His university career was cut short by the war. But anyway, he continued his interest and enjoyed taking me out to the local Warwickshire countryside. I grew up in Coventry, and we used to go pond dipping and looking in streams. So you can see I had a very early introduction to that. But I also realized that my dad was inspired himself by his great uncle. Some of you may have come across William Furneaux. This is a photograph of him in June 1916. And he wrote a book called focusing on the organisms that have lived in ponds and streams, which was published in 1896. So this interest in freshwater ecology clearly runs deep in my family. Well, when I was at school, when I was at school, I got interested particularly in dragonflies. My dad used to take me to a pond near Ashow. Some of you might know it in Warwickshire. It was just a farm pond. It probably doesn't exist anymore, actually, like most farm ponds. And then we also used to go to Ufton Fields Nature Reserve in Warwickshire, which is an old gravel pit, and quickly discovered dragonfly larvae. And I was particularly impressed by them because they basically ate everything that was in the aquaria. I used to have aquaria lined up around, usually they were sweet jars, actually, old sweet jars, lined up around on the windowsill in my bedroom. And I was impressed by the dragonfly larvae eating everything in there. But also, I particularly enjoyed watching the adult dragonflies emerging. Of course, the Ishnids emerged during the night, so I often used to stay up into the early hours of the morning, even on a school day, watching these dragonflies emerge, which then I'd release the next morning. And I relied on Cynthia Longfield's Wayside and Woodland book to identify my dragonflies, and I soon discovered the new Naturalist volume on dragonflies by Corbet, Longfield and Moore, which really whetted my appetite and showed me how interesting this group were from an ecological and biological point of view. So I focused a project, a sixth form, I did biology A-level at school, and my sixth form project was on the dragonflies of Ufton Fields. So, in fact, I actually won the biology prize for that project. I think it's the only prize I've ever won for anything academic, at least. After leaving school, I went to Newcastle University in 1973, where I studied zoology. Norman Phillipson was in the department at that time and ran a freshwater ecology group. Other eminent freshwater biologists were there, including Phil Boone and Ian Wallace and Roger Wootton, who were all studying their PhDs at the time that I was there. And under the guidance of Roger Wootton, I did a project again and looking at what affected the growth rate of dragonfly larvae that I collected in ponds in the Northumberland countryside. When I finished my degree, I did a year doing an MSc, also at Newcastle, in public health engineering. This is an interesting concept in a sense because it mixed biologists who were interested in freshwater ecology with hydrologists who are interested in building sewage works. I'm not sure it's a particularly successful combination. I don't think either group really got into what the other group were doing. But anyway, this is a photograph of me on the left doing a kick sample in one of the River Allens. It's either the East or the West Allen. You can see I had considerably more hair in those days than I have now. And the idea was to look at the impacts of heavy metal pollution on the invertebrates in that river. When I finished my studying at Newcastle, I landed a job at the Natural History Museum in 1979, as Louise mentioned, and I was told to sort out the Kiminoptera collections. And I'm sure, as you all know, as FBA fellows and members, that the Kiminoptera is a large order of insects which comprises Ephemeroptera, Plecoptera, Trichoptera, Odonata, Megaloptera, Neuroptera, and lots of other small orders of insects, which nobody else really seemed very interested in. I think they've probably been looking for years for somebody to look after that collection after Douglas Kimmins retired. So anyway, I beavered away on that collection, mainly actually on the lacewings for the first few years that I worked at the museum. So how did I get into paleo midges? Well, towards the end of the 1980s, the then keeper of entomology, Lawrence Mound, said to me, well, it was about time that I started bringing in some external funding. The museum was looking for new ways to fund itself. And round about that same time, Pete Cranston, who had been working on chironomid taxonomy, left the museum. So there was a gap there. And it occurred to me that if I was going to try and raise money on freshwater insects, midges would be a good group to get into as well. They're clearly a key part of freshwater ecosystems. So I asked Lawrence Mound if he would be happy for me to work on them, and he burst out laughing basically, because he thought, well, they're such a difficult group. But probably if he gives me enough rope, I'll probably hang myself, and then he'd be able to get rid of me and save a bit more money in the department. So he was quite happy for me to try and get into paleo midges. So I was casting around for a while, trying to get to know how to identify them and trying to work out what to do with them, when Rick Batterby came along and gave a lecture at the museum in 1990, talking about the paleo environmental studies he'd been doing on in particular on diatoms, and looking at impacts of acid rain. And during that lecture, he spoke about the other groups of organisms that they found in their lake sediments, among which were chironomids. And he said that there were plenty of chironomids in the sediments, but nobody really knew how to identify them or what to do with them. So that was really a real light bulb moment for me. So I approached Rick after the lecture and suggested that it might be interesting if I took a look at these sediments he'd got and to see what I could do with midges in a paleo environmental context. I've read around the subject a bit more and discovered the work of Ian Walker, who's a Canadian paleo environmentalist who'd been pioneering work on midges in this context. He developed lab methods to extract the midge larval head capsules from sediments, and shown that climate had landscape control on midge distribution. So this sounded particularly interesting to me. So I thought I'd look more into this. The methods that he developed showed that you needed to weigh the sediments. Typically about a gram of sediment would yield 100 plus midge head capsules, which is sufficient to provide statistically robust samples. You deflocculate the sediments by warming them in 10% KOH, sieve them through a 90 micron sieve to extract the head capsules, pick them, which takes a good 5 or 6 hours, and then painstakingly slide mount each individual head capsule. Usually went for 20 per slide under 6mm cover slips. This is a really time-consuming process. Can take at least a day to sort through one sample. And so I was very lucky when Pat Haynes came along. He was an old friend of mine who'd recently lost his job as a graphic designer at BP, actually, but had an interest in natural history. And I knew he was a very careful, painstaking worker. So I asked him if he'd be interested in helping me work on these midges. He asked me how long I thought the job might last, and I said about 6 months. But actually, it turned out to be over 20 years. Fortunately, I did manage to raise money, much to Lawrence Mound's delight, and keep Pat employed as a lab technician working with me on midges for all that amount of time. So I mentioned that I'd had a chat with Rick, and he had a project running with Helen Benyon and Don Monteith in his lab at UCL and on the Shropshire and Cheshire Meres. So this is a Ramsar site, which is currently eutrophic, and there was some doubt about whether or not these lakes were naturally eutrophic or had become eutrophic in more recent times. So the project was designed to have a look at that. And so we collected a whole load of sediment cores, one in particular from Betton Pool, which I had a look at for midges. And these are the results that I got. Unfortunately, I'm going to have to show you a few more graphics a bit like this. But what you've got here are the midge taxa along the top and their relative abundance. So the wider, the thicker the black line, the more abundant each taxon is. And then down the side, we've got depth with surface sediments at the top and going down, in this case, to 90 centimeters. And the core has been dated with lead-210 dating, which shows us in quite a lot of detail that the core stretches from 1995 down to beyond 1857, which is beyond the mid-19th century. Lead-210 dating doesn't work really. But anyway, what you can see from this this diagram is that there actually has been a big change in the midges, and there's declines So taxa that were present prior to the mid-1940s, which are typical of mesotrophic or oligotrophic waters, decline, and there's a big increase in taxa after the mid-1940s, such as chironomid taxa in the main, Glyptotendopes, Chironomids, Endochironomids, which show big increases. And so this suggests that actually these lakes aren't naturally eutrophic, and they've become eutrophicated since the mid-1940s. We actually took a whole load of surface samples as well from 44 lakes, covering a range of total phosphorus concentrations, and developed an inference model. So you can see here, this is observed total. These are the lakes, the 44 lakes plotted here against the known total phosphorus at each one of those lakes, and then the predicted total phosphorus based on a midge inference model. So we worked out the total phosphorus optima for each midge taxon, and then used a weighted averaging to develop that, and then using leave one out, so you can estimate the midge inferred total phosphorus of the lake using all the other lakes in that training set, the other 43 lakes, to infer that, and then compare the inferred total phosphorus based on the midges against the known total phosphorus to develop this inference model. So you can see that it's a relatively good it works reasonably well. But the problem is, of course, is that midges aren't responding directly to total phosphorus, whereas diatoms are, which is why diatoms work much better in this sort of model. But when we plotted the midge inferred TP against diatom inferred TP, you could see there's a fairly, well, pretty good actually, relationship between the two. So midges do seem to work reasonably well in these sorts of lakes, shallow lakes, to infer TP. So that was an encouraging start to my work in midge paleo environmental studies. But the main interest I wanted to focus on was climate. And Rick also introduced me to Brigitte, a man who is based in Bern in Switzerland, who's working with Herb Wright, and also John and Hilary Burks, who are also working with Brigitte to look at impacts of climate change on Swiss alpine lakes. And in particular, we were looking at Lake Gertzensee, which is about 600 meters above sea level, and Lake Lausanne, which is about 1,200 meters above sea level. And I was introduced to the arcane work of coring lakes from peat bogs. You can see here 4 people required to try and heave that corer out of the lake, out of the sediments in that peat bog. The lake glacial lake was about, I can't remember how many, several meters below ground. So these are the results I got from Gertzensee. I'll just show you the Gertzensee, and they were very encouraging. They actually reflected the sort of results that Ian Walker was getting in Canada. And here's the lake, so 170 centimeters below the surface of the bog. Was about 1,100 years ago. This is using radiocarbon dates, and the sequence went down to about 1,300 years ago. And because of the dates, we knew where we were in the sequence, and we knew the Younger Dryas was in this part of the core. And what I want to point out is, so here are the different midge taxa which have been grouped according to their thermal optima, and you can see in the Younger Dryas that the sequence is dominated by cold water taxa, whereas warm water taxa are eliminated during the Younger Dryas. So there's a clear climate signal in that sequence. And I remember this particularly impressed John Burks. He thought there was a lot of potential in midges as climate indicators. The point is really that the midges are directly responding to climate in a way that a lot of other proxies that are used for inferring climate change aren't. They're much more indirect in their responses. So John then invited me to join a project that his wife Hilary was running at on a lake in Crocodiles in northwest Norway. You can see the lake in the background here. And it was a big multi-proxy project. The idea being that the more indicators that you have in examining in these lake sediments, the clearer the idea you have of how climate is impacting these organisms. So we have Geoffrey Lemdahl here, who's looking at beetles. There's Rick looking at diatoms. Dave, I can't think of his second name. Anyway, Dave was looking at stomatal densities in leaves. John Small's there, another diatomist, and works on Cladocera. Ingrid Solhoy was looking at mites. There's Bert Herb again, and there's me in the background. I was the only midge person on that project, and there were also morphologists and radiocarbon experts as well. And this is the lighthouse at Crocodiles, which we all stayed in. So had a look at the cores from Crocodiles, and again got very similar results to what we were finding in Switzerland as well. So here are the taxa along the top again. This is depth going down through the core, and again you can see cold water taxa. Oh, and the radiocarbon dates here. So we know this is the Younger Dryas, centered around 1200 years ago. No, 12,000 years. 12,000 years ago. Yes, that's what I meant, not 1200 years ago. Excuse me. Anyway, so you can see the cold water taxa dominating in the Younger Dryas, whereas the warm water taxa, again, eliminated during this cold period of climate. Around about the same time that I worked with John and Hilary at Crocodiles, Rick was involved in a grant from NERC called Tigger. And again, this is a lot of people were involved in this, looking at climatic change and environmental change from modern day right back to the late glacial. And also involved in that project was a group led by John Lowe at Royal Holloway, who was particularly interested in the late glacial. The reason climatologists are interested in the late glacial is because it's a period of this very rapid climate change and rapid oscillations in climate, switching between cold and warm periods. So it's a very good, clear idea, or potentially clear idea, of what is driving climatic change, but also how fast organisms respond to climatic change. So John had a core from Wittrig Bog in Scotland, which covered the late glacial sequence. And I persuaded him that it would be a good idea to look at midges from this core. He decided to take a punt on that. I didn't have much of a track record, and nor did midges really, for that matter. But I think it was quite a generous grant, actually. So he decided it was worth getting me to have a look at the midges to see what we could get out of it. And again, you see the same similar story where during the Younger Dryas, we have cold water taxa dominating the sequence, and these temperate taxa, warm water taxa, being excluded. So clearly a very cold period. But what was interesting from this core, which hadn't really emerged so much from some of the others, was evidence for midges responding to other climatic shifts earlier in the sequence. So down here, at this level down here, around about 225 centimeters, you can see there's a resurgence of cold water taxa, and warm water taxa are also being eliminated. And another part of the sequence here at about 185 centimeters, more cold water taxa coming in, and some of the warm water taxa being eliminated. But clearly these cold water episodes, or cold climate episodes, are of different intensity. So in the Younger Dryas, this is obviously the coldest part of the sequence, where there's a huge domination by cold taxa. Down here, fewer of their warm water taxa are excluded, and not as many cold water taxa come back. And in this one, even fewer are affected. So it seems from this that this Younger Dryas is the coldest part, then this is the next coldest part of the sequence, and then this the next one. So there's basically 3 different intensities of cold climate within this core, separated by warm water periods. So from this we can see that chironomids have got several attributes which make them suitable for paleoenvironmental analysis. First of all, they're ubiquitous. They're pretty well present in any lake sediments you look for, except for deep water and anoxic lakes. They're very abundant. In about a gram of sediment, we can expect to find at least 100 head capsules. They're also identifiable. It's the head capsules that are preserved in the sediments, and most of the larval taxonomy is based on features in these preserved head capsules. They're also species rich. So each taxon has a different optimum for different parts of the environment. So some might be very sensitive to acidity, others to climate, and so on and so forth. So you can build up a very detailed picture from the midge assemblage of what's going on in the environment. They also complement other organisms that might be used in these climatic studies, like diatoms, for example, which tell you what's going on within the water column, the water chemistry, or pollen, which might be telling you something about how the surrounding landscape of the pond is being influenced by environmental change. The midges also respond very rapidly to climate. Of course, the adults are winged, so they can cross the landscape very fast, often wind assisted. And so they can arrive at lakes very soon after the climatic's changed. A lot of the taxa have very narrow tolerances to particular elements of the environment. They're stenatopic. So that means, again, that is a major attribute for midges in terms of understanding how the environment, how they're responding to the environment, but also in terms of developing quantitative models using midges. Very key point is that we know the midges have developed in situ in the lake. There's no issue about long distance transport. In fact, the head capsules of the larvae that we use in these analyses are derived from the shed skins of larvae. They're not dead larvae. So we know that the larvae have successfully completed their development in the lake in conditions that they're finding suitable. There's no problems with long distance transport, for example, which might get with pollen, which is blown around in the environment and may be derived from a long way from the sediment cores that you're actually examining. So having built up this picture of midges responding to climatic change, John Burke suggested to me that it would be useful to develop calibration sets based on midges in order to reconstruct climate based on these fossil assemblages. So John had already developed a calibration set for pollen across Norway, and he suggested he'd got surface sediments left over from his pollen data set, and he suggested to me that I look for midges in those surface sediments and began to build up a midge calibration set. And we also added lakes to further enhance that calibration set from areas where there were gaps, basically, in his pollen data set. So we actually sampled new lakes in central Norway, in the high alpine region of central Norway, and also in the far north of Norway as well. And we developed this calibration set, which part of it, this is about half of it I'm showing you here. So what we've got here are the midge taxa along the top, and each one of these lines is a separate lake, and they're ordered according to the mean July air temperature of each one of those lakes. So you can see here at the left, taxa like Sectrocladius semicirculatus, Microsectra bidentatus are dominating in warm water lakes in the south of Norway. So we had about 100, well, we did have 157 lakes stretching from the far north in Svalbard, where mean July air temperatures are 3.5 degrees centigrade, right down to the far south of Norway, where mean July air temperatures are 16 degrees centigrade. So we have a very wide temperature gradient to develop this training set. And you can see as the lakes get colder, different taxa start to dominate the assemblages. And this is the other half of the training set, where you can see here in these ultra cold lakes in Svalbard, which are dominated by just a few taxa actually, like Microsectra radialis and Hydrobenus. So this provided us with a really good data set in order to develop what we call a Midges Thermometer. So again, like the TP training set I showed you earlier, we've got this is the model here. So we've got observed mean July air temperatures of each one of these 157 lakes plotted here against the predicted mean July air temperature based on the temperature optima of each one of the Midges taxa, which we derived using weighted averaging from the whole training set. And again, use this leave one out cross validation. So you estimate the temperature of one lake using the midge assemblages from all the other lakes, and that gives us the model. And you can see the model works a lot better than the total phosphorus model. The R squared is about 0.9, and the root mean squared error of prediction is about 1 degree centigrade. So the model works very well. And this is because midges are directly responding to the climate. We used air temperature, you'll have noticed, rather than water temperature, because it's much easier to derive air temperature data from the numerous weather stations scattered around Norway, rather than trying to measure water temperature, which would have taken years. It's probably impossible, actually, using data loggers, for example, in the lakes. And we know that the air temperature and water temperature of lakes, in most circumstances, is linearly and very closely related. So we applied this inference model to the Wittrig bog sequence, and you can see we got this very nice temperature inference. So you can see air temperatures, these are mean July air temperatures increasing very rapidly at the beginning of the late glacial, and then gradually cooling, punctuated by a series of cold oscillations, and then again air temperatures declining extremely rapidly into the start of the Younger Dryas, and then through the Younger Dryas gradually increasing with again a few temperature oscillations before at the beginning of the Holocene the temperature really rapidly increasing again. And we compared this with the Greenland ice core data based on stable isotopes. The stable isotopes are the ratio of oxygen 18 to 16 is related to temperature. So you can derive a temperature curve, although there's no degrees centigrade applied to this, but it gives you a relative temperature curve. And you can see in general that the Greenland ice core record, which is really the gold standard that's been used to tell us what at least climate change is doing in the North Atlantic, compared with the Wittrig bog. But there are subtle differences. So you can see that during the interstadial here, the temperatures are declining very rapidly in Greenland ice core record, whereas it's much more gradual decline at Wittrig bog. And also these two oscillations here, 1D at Wittrig bog is much colder than oscillation 1B, whereas the reverse is true in the Greenland ice core. Whereas other parts of the sequence are very similar. For example, the gradual warming in the Younger Dryas. So I was intrigued to know whether, well, it looked like, first of all, that the inferred temperatures we were getting from the midges made sense. This was something real happening. But were these differences reflecting real regional climatic differences or not? Was there some error, basically? So over the years, we built up a whole load of several other sites in Scotland, Northern England, and in Ireland, and actually compared these with Wittrig bog and actually found very, very similar temperature evolution in the interglacial that we got at Wittrig bog. which led us to believe that actually that pattern of temperature development in the interstadial, which was different to the Greenland ice core, was actually a real local difference driven by local climate forces. I won't go into all the details of this figure, but you can see, roughly speaking, we've got a very similar temperature trends. Now, an ambition of mine for a long time had been, so Russell Coop, who'd been working on beetles in these similar sequences and used beetles to infer temperature change, had developed isotherm maps for Northwest Europe based on beetle inferred temperatures. And it had been a long-held ambition of mine to develop similar isotherm maps for chironomids. It took us a long time to get there. So Russell had published his data in 1998, and it wasn't until 2014 that I and Pete Langton managed to assemble enough data. But lots of other people have been working on sites throughout Northwest Europe. There's over 20 sites here. But it just emphasizes how long it takes to develop these data. And we built these isotherm maps and then compared with them with the beetle data. So in the top left-hand corner are the isotherm maps for the beetles, and in the larger maps are the isotherm maps based on the chironomids. And again, I'm not going to go into the details of these, just to say really that the temperature developments indicated by the beetles and the midges are very, very similar. But there is a big difference. And if you look carefully, you can see there's a lot more warm colors in the beetle isotherms than the chironomid inferred isotherms. And in fact, the beetles, which we found estimating temperatures pretty well consistently +2 degrees higher than the midges. Now, it's very difficult to say which one's correct, but of course, I'm going to say, I suspect the chironomids are giving us a more accurate idea. I think the reason the beetles may be overestimating by about 2 degrees is that they can shelter in microclimates in a way that the midge larvae can't. So as the temperature cools, the beetles find small areas in which the microclimate is slightly warmer than basically the ambient temperatures, whereas the midges living in a lake, there are essentially no microclimates within. Obviously, the littoral and the profundal will have different temperatures, but that's as far as you can get with microclimates in a lake. So I think that's probably the reason for this difference. While I was doing all this work, Oliver Hayrie, based at Bern University, was also developing calibration sets based on midges in Switzerland. And we decided it'd be a good idea to merge the two training sets, the Norwegian and the Swiss training sets, to develop a pan-European chironomid-based training set, which could be used on sequences throughout Northwest Europe. And this is the model we developed. You can see with Andy Lotter and John Burks again, you can see that this is the Swiss training set with open circles, has warmer lakes, so it expands the training set at the warm end, whereas the Norwegian training set expands the Swiss training set at the cold end. So we've got a longer temperature gradient. There are far more lakes. There's over 200 lakes, nearly 300 lakes in the training set. So the R squared isn't quite as good. The root mean squared error of prediction isn't quite as good, but it's still a robust model. And one of the things we did with this, so a lot of the time people say, well, why are you doing these reconstructions with midges? And one answer is often, well, it's to test climate models. This is actually very rarely done, but we did get the opportunity to do this working with Hans Rensen. And we've compared midge-inferred temperatures across a whole series of sites, well, the same, but essentially the same, a lot of the similar sites that I showed you that worked on with Pete Langdon, and compared the midge-inferred temperatures from those sites across Northwest Europe with the results of this climate model called ECAM. And basically, the results are very similar, which is, of course, encouraging. There's, you can see there's a decrease in the warming across from west to east as the climate warms right at the beginning of the interstadial. You can see this is the Younger Dryas, early Holocene transition. And what you can see here is in mid-latitudes here, the warming is much stronger than in high latitudes and low latitudes. But the other thing to notice is that the model appears to be overestimating temperatures relative to the midge inferred temperatures, which you can see in particular in the warm phases of this sequence, of this period. So you can see that in these two figures on the right. The bottom one, showing the warm phase, shows these model inferred temperatures higher than the midge inferred temperatures. So it does appear that ECAM is overestimating these warm periods. And so Hans went off and adjusted his model accordingly to get it to fit better with the midge inferred temperatures, because of course it's very difficult otherwise to validate these climate models. The best way to do it is to try and infer temperatures from the past where we know what the temperature was. And of course, during the late glacial, as I've mentioned, because of these very rapid and strong temperature fluctuations, it gives a good idea of what climate forces are in operation at that time. So having done these models in Western Europe, thought it was a good idea to try and develop models in other parts of the world in order to try and use midges to infer climatic change as far across the globe as possible, really. So I worked with Angela Self and Larissa Nazarova, and also Viv Jones and Nadia Soloviva at UCL to develop midge calibration sets across Russia. So you can see these are the sites that we developed. There are about 250 lakes, I think, altogether across on this training set, which we used to develop a midge calibration set. You can see we arrived in helicopter in the Putaran Plateau for a very adventurous field trip. And Angela developed this model, and one thing that came out of it, which I thought was interesting, was the effect of continentality on species optima, which I'm showing here on the left. What I hope you can see is these are the different taxa along here, and these are their temperature, weighted average temperature optima. And you can see that the temperature optima of a species in Siberia, for example, is pretty well consistently 2 degrees higher than the temperature optima of the same species in Norway. And I think this is due to the effect of continentality. The Siberian summers are much shorter, and so for the midge species to complete its development successfully in that shorter growing season, it needs to have summer temperatures at least 2 degrees higher than are needed in Norway, where the summer's much longer, or relatively longer, and so the midges can complete their development. So this made us realize how important it was to apply calibration sets from to sediment sequences from the same region that they were collected. But also because the temperature optima might vary from one part of the world to another, even of the same species, because of climatic effects. But also it enabled Angela to develop a continentality training set, which was quite useful because there's been a lot of discussion about how continentality might change during different climate regimes. So it was expected, and we found that in Northwest Europe, the climate was much more continental during the late glacial than it is today. I worked with Will Gosling and Fraser Bird and Joe Williams as well at the Open University to develop a calibration set based on midges from the Andes, which we also applied to some sediment sequences. Now, the midge larval taxonomy of midges is pretty poorly known in Western Europe when I started working on this, which meant that I had to develop my own sort of taxonomic approach to working on these midge samples. But in the Andes, of course, the midge larvae are even more poorly known. So it was quite an uphill struggle for me, as well as Fraser and Joe, who are the PhD students at the time, to develop a taxonomic system that was robust and reliable. I also worked, again, through contacts that Rick put me in touch with people at the University of Lanzhou who were interested in developing midge taxonomy and paleo research based on midges in China. And a couple of students came to work with me at the Natural History Museum, Zhanhui Chen and Haipeng Wang. And their lakes were basically in the arid northwest of China, where climatic change is mainly driven by shifts in the monsoon. And so it was reflected in precipitation rather than climate, and effectively in the lakes, how saline the lakes were. And this is a sequence Zhanhui had from Sugan Lake. And he came to work with me for a year, and it quickly became apparent when we looked in his core that essentially there are only 4 taxa. And I was very worried, actually, that we wouldn't get anything useful out of it. But he put in a lot of hard work. You can see how many samples he processed over that period of time. And it actually worked out very well, because 2 of the taxa were tolerant of saline conditions, whereas the other 2 taxa were tolerant of freshwater conditions. And they switched from one to the other as the monsoon shifted. And this is the, he used a salinity, a chironomid inferred salinity training set, which had been developed by other colleagues in China, and produced this sequence here of inferred salinities. And you can see during this is the Little Ice Age, this sequence again dated by lead-210 dating, you can see that the lake is less saline, and also there's much higher fluctuations in rainfall during that time. And also there's another period here about 1200 AD where there's another part of the sequence where the lake is a lot fresher. And this reflected shifts in the monsoon over that part of the world. So again, showing the sort of flexibility and of different aspects of the environment that chironomids can be used in to investigate. So that's the paleo stuff that I was doing, but I just wanted to sort of finish off by talking about other work that I've used midges for. And I came across a paper by Doug Benn, who was describing fluctuations in the productivity of the Slavonian grebe, which is shown in the bottom left here. And he had annual data from Loch Ruthven. Loch Ruthven's a loch, is Inverness. Loch Ruthven's just down here. This is Loch Ness. And Loch Ruthven is the main breeding site for the entire population of Slavonian grebes in Britain. And he showed that there was massive fluctuations in the productivity, how many chicks each pair produced from year to year. But they weren't sure what was driving these fluctuations in productivity. But when I looked at the data, you can see this is the grebe productivity here in the blue. It struck me that it could be climatically driven, in particular from fluctuations in the North Atlantic Oscillation. So I contacted Doug and asked him if it'd be okay for me to core the lake and see if there was any link with midge abundance, or in fact whether I could infer climate change from the midges. So we took a sediment core from the lake and we sliced it very finely at 1/4 centimetre intervals and got that dated, again using lead-210. And it showed that each slice, each 1/4 centimetre slice was round about 1 year. So I was able to compare on an annual basis the midge abundance with the grebe productivity. So the pink line here shows the concentration of larval midge head capsules per year against the grebe productivity. And you can see there's a pretty, well, it is a significant match, especially when you adjust for errors in the age model. So it does look like the midges are, well, either the grebes are affecting the midge abundance, which, considering there's only a handful of grebes on the lake, seems highly unlikely, or it's the midge abundance that's driving or linked to the grebe productivity, which I think is much more likely, because when we consider that the adult midges are emerging from the lake just at the time that the grebe chicks are hatching, and then they can be feeding on adult midges as they emerge from the lake. But then the question is, well, what was driving these midge fluctuations? So it seemed to me that productivity could be something to do with it. And this is showing a plot of loss of ignition. And so essentially, this is a measure of carbon in the lake sediments. And you can see that midge abundance tracks very closely the loss of ignition curve in these lake sediments, which suggests that, oh, and the other point is that around about 1940, there's a big increase in loss of ignition, which could reflect changes in agricultural practice around the lake. But whatever it is, you can see lake productivity goes right up, and that's tracked by midge productivity. Viv Jones also did a diatom inferred TP reconstruction from the same sediments, and you can see again that the midge abundance is tracking the inferred total phosphorus from the lake. So it does seem like lake productivity is driving these fluctuations in midge abundance, which in turn are driving the productivity of the Slavonian grebe. Now, I was hoping that I'd be able to link these changes with perhaps changes in rainfall, or as I mentioned, the North Atlantic oscillation, but I couldn't find any significant relationship. So that question's still hanging. But this interested me in looking at how midges could further look into bird conservation questions. And Hannah Robson, we got to know Hannah Robson, and she was interested in this study of Slavonian grebe. And so under Viv Jones and my supervision, Hannah worked on the common scoter, which is it's a marine duck which breeds in a few lakes in northeast Scotland in the flow country. And she took out a big Bencora, which Rick's group at UCL had developed. This is a 14 centimetre diameter cora, so much bigger than the cores that we would typically using. But it means you collect a lot of sediment, and so it enabled Hannah to look at not just midges, but also Cladocera, diatoms, mollusks, other inverts, beetles in particular, macrophytes, and also fish, to try and build up a picture of how the entire ecosystem in these lakes is working and how that might influence the productivity of the common scoters. And I'm not going to summarize all the data, just show you the midge data here. And what you can see is in the mid-1990s, there's a big shift in the midges, and you get big increases in most taxa, apart from the diamesines, which is indicative of the lakes becoming more eutrophic. There's no evidence for impact of increasing DOC or fish or acidification on the scoters, which are all things that had been suggested could be affecting scoter breeding. But so it looks like there is some link with eutrophication of these lakes and the scoters not doing quite so well. Another recent interest I've had is looking at how, so we know that basically there are three responses of organisms to climatic change. There are shifts in distribution, there are shifts in phenology, and there are shifts in size. And so I was interested to see if we could use the museum collections to look at least those second two questions. So I started looking at butterfly phenology over the last 150 years based on music collection dates of museum butterflies. We had tens of thousands of butterflies in the collection, and showed that the butterfly phenology, pretty well all of the species, was advancing compared with climate data from each year that they were collecting. But we were also interested in testing the temperature size rule, which states that organisms should get smaller as the temperature increases. The way that this applies to insects is that increasing temperature accelerates their development rates, and so the adults emerge earlier than they would in cooler temperatures. But of course, the larvae haven't had so much time to grow, which results in smaller adults. So Rungtip Wonglerslack looked at both midges and dragonflies to see if this held for those two taxa, and she used the mesocosms at the FBA lab in Wareham, where she put you can see her emergence traps here. She floated emergence traps on 10 of the mesocosms that were running at ambient temperature and 10 mesocosms that were running at +4 degrees above the ambient temperature, collected all the midges throughout the whole growing season that were emerging from the lakes, and then measured their wing length. And of the species she could identify and also had sufficient numbers of, in almost all of the cases, the midges emerging from the ambient temperature was significantly larger than midges emerging from the heated tanks, which backed up the temperature size rule. Interestingly, in the dragonflies, she got similar results with damselflies, but not with territorial dragonflies, in particular libellulids and ischnids, where there didn't seem to be the temperature didn't seem to be affecting them. So the reason for this is, we think, is that because they're territorial, it's very important that the larger the individual, the more successful it will be at holding its territory. So it's essential that they maximize their size. And so this accelerated development because of warming is outweighed really by the need for the adult males to be as large as possible. So just to summarize, I hope by now you can see that midges can be popular. I'm just showing you here, this is a midge workshop of palaeo-ecologists. I'd like to stress here, I don't think there are many biologists among this group. So all these midge aficionados started off on the whole as geographers and have embraced midges in a way that a lot of biologists or freshwater ecologists haven't. Anyway, so they can be popular. They can also be very useful, in particular for looking at climate change and past climate change, but also for addressing questions on modern ecology and of interest in bird conservation, for example. Contrary to what a lot of people might tell you, they are actually relatively easy to identify, and this has been simplified by the hard work of several palaeo-ecologists who have worked on midge larval taxonomy in order to be able to identify their samples. And with Pete Langdon and Oliver Hayrie, I produced this book on the identification of Palearctic chironomid larvae, which is, of course, very useful for modern ecologists as well, which was published by the Quaternary Research Association, again, an organization made up mainly of geographers and palaeo-ecologists. It's actually their top-selling guide and one of my highest citations as well, 648 citations to date. So they are popular. Charismatic, well, I think probably I'd grant you they're not particularly charismatic, and of course they don't bite. One thing that the FBA might consider is a midge field guide. Several of my students, particularly Murray Thompson, who I'm going to come to in just a moment, was able to sort his midges by sight, well, you know, under a low-powered microscope, just by looking at the relative size of their heads, their colour, relative size of the head to the abdomen, and so on. He could sort them out by eye, and then check the identifications under the microscope, having mounted them, slide mounted them, and confirm that a lot of them are actually identifiable to species just through low-power microscopy. So there's something to ponder. So what next? Well, as Louise mentioned at the beginning, I'm still heavily involved in the Riverfly Partnership, which currently is hosted at the FBI. I'm currently chair of that, where we train volunteers to undertake biomonitoring of rivers. This has proved very popular. We've got over 3,500 volunteers across the country, sampling about 2,500 sites on rivers, mainly in England, but also in Scotland and Ireland and Wales. They're sampling these rivers every month, so much higher frequency than the Environment Agency are currently able to do. So keeping a careful eye on water quality in those rivers. I'm also interested in river restoration. This interest really started with a project I worked on with Carl Sayer at UCL and Murray Thompson, who was our PhD student, who was looking at how important putting large woody debris into rivers was in restoring those rivers to much more natural systems. And what he actually showed is in fact the midges that show the biggest response in terms of abundance and diversity changes once the woody debris has gone in. And I'm also interested in pond restoration. Again, working with Carl at UCL, and we had a student, Jonathan Lewis, who recently finished his PhD, who was able to show how important restored ponds on farmland were for restoring not just aquatic biodiversity on ponds, but also to support and increase the diversity of farmland birds. And again, the midges come in here because it's primarily the midges which are emerging in large numbers from these farmland ponds, only after they've been restored, that is, and supporting this high diversity and abundance of birds. So I've been working recently with a large landowner in Hertfordshire, and his wife have started to restore some of the 30 ponds on their farm. And you can see one of them, so they've done 3 this autumn. This is the first one, before, above, and after, below, letting a lot more light into the pond. And hopefully we'll see the diversity of organisms in the pond increasing, but also more birds using the pond as well, and increasing the bird populations on his farm. And we're hoping to encourage other landowners in Hertfordshire to also work on their ponds and develop a Hertfordshire pond project. And what else what I'm planning to do? Well, I've got more interested in birds. I was interested in birds years ago, but set myself a target of trying to see all the breeding bird, well, not just the breeding birds, but the common migrants as well. I've got quite a long way towards that target, but I've still got a few on my list. I'm hoping to tick off in North Norfolk this winter, for example. I also keep my hand in with dragonflies, but also I might turn up at a pub near to you with my Morris dancing side as well. You can see me here on the right playing melodeon. I have been known to set foot on the dance floor with my clogs as well. So you never know, I might show up at a pub near you. Okay, so that's all I wanted to say. I'll stop the share and come back to you.
Thanks, Steve. That was brilliant. I think we're a little bit over time, but I think we've got a couple of questions that have come in. Okay. So the first one was from Helen James. Helen, would you like to unmute yourself and ask Steve your question?
Hi, Steve. I was just wondering actually about the identification of the species in the paleoclimates to the modern ones and how they vary. I mean, are they the same species that you were seeing, or are they different species? And thanks, by the way, for a fantastic talk. I really enjoyed it.
No, thank you. So, okay, so when I said we can identify them to species, that was a bit shorthand. So what we can do is identify them to species groups. So in some cases, that might actually be only one species, but in other cases, it could be several species. But actually, that's one of the, as far as you can often get with modern specimens as well. Having said that, I'm pretty sure that we are looking at the same taxa, because the same collections of taxa that we get in a modern cold lake reappear essentially in cold periods in the past as well. So I think their ecologies haven't changed, at least over the last 15,000 years. Mm-hmm. Having said that, I have looked at sequences from a long time ago. So the earliest one I looked at is about 450,000 years ago. And there were suggestions in that sequence that things might have changed. So there were 1 or 2 taxa that I didn't recognize. I could tell which genus they were, but they didn't look like any of the species groups I'd seen in that genus before. But also, they're compared with the beetles, which we had from the same site. So we knew from beetles what the temperatures were. They seem to be occupying a wider temperature niche than they might be expected to today. So I think going that far back, there could be differences.
Great, thanks very much.
That's great. Thanks, Steve. I had a question. So you said that that midges are obviously great food for other species.
Yes.
So I was just wondering whether, can you predict from your models what human-induced climate change might be doing with midge distribution and how that might affect food webs in future?
Okay, so I think it's probably unlikely that it will affect food webs, because basically, as the climate warms, we'll just get a shift in the diversity of taxa in these lakes. But in terms of abundance, they always seem very abundant. And in fact, the warmer it gets, the more abundant they seem to get. So, and obviously they're a really important part in food webs. I think the only problem will be is if these lakes become more depleted in oxygen, but then that's going to affect all taxa anyway. But even then, you're likely to get masses of, chironomids, for example, the bloodworms will build up in massive. So simple answer to your question is I don't think it will affect food webs, because midges are always going to be there in one shape or form, I think.
Cool. Excellent. Thank you. I can't see any other questions. So yeah, I guess all that's left to say is that's been a brilliant talk, Steve. Thank you.
Okay, thank you.
And yeah, we're taking a break from our FBA seminar series over the Christmas and into the new year. So we will post details of our talks in the new year. I believe our first one is on the 2nd of February. So we will post some more information on the FBA website soon. So keep your eye on that. And thanks again, Steve.
Okay, my pleasure. Cheers.
Thanks very much.
Okay.
Bye, everyone.